My name is Brandon Bell, and this is exam mini video 5. The adapter theory was used by Francis Crick in order to propose the function of tRNA in cells. This theory explains how information DNA specifies an amino acid sequence of proteins. This theory revolves around how information can be extracted from nucleic acid and used to put together amino acids in a specific sequence, which in turn determines the nucleotide sequence of the nucleic acid. Crick had believed each amino acid attached to its own specific adapter piece of nucleic acid. These adapters help incorporate amino acids to a growing polypeptide. We now know that these adapters are known as tRNA molecules. The nucleotide sequence of tRNA molecules creates this plastic cloverleaf of tRNA molecule, which is a secondary structure of tRNA due to base pairing. They are secondary structures because they are not linear, but they are rather folded and have a second level of structure. Here's a cloverleaf model of tRNA. The mature tRNA has unusual modified bases. At the top is where the amino acid is attached. At the bottom is the stem loop structure of the anticodon, which contains 3 complementary bases that will base pair with the mRNA in order to bring with it the appropriate amino acid, and then one of the base pairs in the anticodon also happens to be modified. Crick believed small RNA molecules would be the adapters that would be able to be charged with amino acids by specific enzymes and would be capable of identifying the codons of the mRNA by base pairing. tRNA is involved with transferring an amino acid to a growing peptide chain by reading the RNA-mRNA sequence. tRNA carries amino acids to the ribosome, and they act as bridges matching a codon and an mRNA with the correct amino acid it codes for. The anticodon region of the tRNA will base pair with the mRNA and bring with it the correct amino acid. tRNA mediates the translation of mRNAs on ribosomes due to the universal genetic code. Also, tRNA helps in the incorporation of the amino acid into the growing polypeptide on the ribosome. The universal genetic code is where a specific codon in the RNA always specifies a specific amino acid in the protein. A proposal for an alteration that would prevent tRNA from acting as an adapter that follows the universal genetic code would be if the anticodon were to be complementary to the stop codon. A normal translation occurs by the ribosome and would terminate when it reaches the stop codon. However, a mild alteration of the anticodon being complementary to the stop codon would create many consequences for the cell. Rather than termination occurring, translation will continue to occur and no protein will release. And then these are my references, and thank you.